Welcome to Better Health Guy Blogcasts, empowering your better health. And now, here's Scott, your Better Health Guy. The content of this show is for informational purposes only and is not intended to diagnose, treat, or cure any illness or medical condition. Nothing in today's discussion is meant to serve as medical advice or as information to facilitate self-treatment. As always, please discuss any potential health-related decisions with your own personal medical authority.
Hello, everyone, and welcome to episode number 60 of the Better Health Guy blogcast series. Today's guest is Dr. Eva Sapi, and the topic of the show is Borrelia research. Dr. Eva Sapi is a professor and department chair at the Department of Biology and Environmental Science at University of New Haven, where she teaches undergraduate and graduate biology courses and carries out Lyme disease research with her graduate students. She is the director of the Lyme Disease Program. Dr. Sapi graduated with a master's in genetics and molecular biology from Eotvos Lorand University in Budapest, Hungary in 1987. She received her PhD in 1995. Her research interest is to investigate the different forms of Borrelia burgdorferi to better understand how Borrelia can hide from the immune system as well as from antimicrobial therapies. Her recent research shows that Borrelia burgdorferi is capable of forming a protective layer around itself called biofilm, which could render it to be very resistant to antibiotics and provide a logical explanation as to why extensive antibiotic treatment for patients with a tick bite history could fail. The goal of her research group is to fully characterize this novel form and to identify novel antibacterial agents that are effective in killing all forms of Borrelia burgdorferi. Dr. Sapi organized and chaired 6 Lyme Disease Symposiums at the University of New Haven during the last several years. Her research interests include Lyme disease, pathogenic biofilm, antibiotic resistance, and the different morphological forms of Borrelia burgdorferi. And now my interview with Dr. Eva Sapi. So I have had the honor of knowing Dr. Sapi for probably a decade since we first met through the iLads events, and I've always found it comforting to know that people like her are out there trying to find better solutions for those of us that are dealing with Lyme disease. And so it's an honor to have her on the show today. Thanks so much for being here, Dr. Sapi.
Thank you, Scott.
So tell us a little bit about what drew you into the field of researching Lyme disease. Have you had any personal experience with Lyme disease yourself, or was it just something that one day you decided was your interest and passion?
No, it was very much more exciting than that. I was a cancer researcher at Yale, and I was looking for a job and a professor position, and I got the professor position in 2001, and very, very soon after I came down with Lyme disease, and it was horrible. It was Lyme, so you could imagine. And the worst part of it, I just got this new position. I was so proud of it, and I felt like I'm losing the position, I'm losing this job, and just I couldn't teach. Can you imagine teaching with Lyme?
No.
I was petrified in a classroom. I was reading my slides and just surviving those classes was horrible. So yes, I had personal experience with Lyme.
So can you tell us a little bit about the life cycle of Borrelia, and why does the long duration of its life cycle matter when we consider possible antimicrobial therapies?
See, I think we still need to understand the life cycle of Borrelia. Right now, we always think about the spirochete, yes? And when we do any kind of antimicrobial experiment, we focus on a spirochete. Just very recently, we start to look for called the persisters, which is usually like aggregates of spirochete. But actually, all lab was decided that it has to be more forms which can be resistant to antibiotic. And we remember that in 2012, we published a paper that this bacteria might make biofilm. And why was it important? Because biofilm is known to be resistant to antibiotics. So since then, our group is testing all the forms of Borrelia. So we believe spirochete is probably the form which is very transient. It just has one function to sort of move around. But when the infection established, is more like in biofilm form, when it's like nicely create these little biofilm cities and protect themselves from everything.
And so the fact that the life cycle, as we have thought of it historically, is fairly long relative to other bacteria, is that an explanation for why treatment of Lyme disease often also takes a long period of time?
I mean, obviously that question is still people fighting over on this question. What we're seeing in the lab is when we're treating in the lab in a culture tube, we're treating Borrelia. It takes more than usual. You would treat like E. coli or some other bacteria to really eliminate it. Even when we think we eliminate it, when we call the recovery culture, when we remove the antibiotics, it's still most of the time it's coming back. So it's not just about that, how fast or how slow it grows, but it has these other forms when it can survive in it, and after it can come back weeks, months. We had one experiment when back in 2005 or 2006 when we had some cyst form, and we tried to see if we can bring it back, and took like weeks and weeks to bring them back fully to spirochete forms.
So historically, we've talked about the spirochete, we've talked about the cell wall deficient or L form, we've talked about the cyst form. So is the persister form different from the cyst form of Borrelia?
What we call persistence right now, we call the stationary culture. And you're going to say, what is stationary culture? Stationary culture, when you grow Borrelia and it started to grow, and when it reaches a certain density, it starts to make aggregates. And when these aggregates form, something happens, because definitely at this point they are way much more resistant to antibiotics. Now, these aggregates actually can settle, yes, and on some solid surface, which could be could be some tissue, or could be just a simple. We had the experiment, even glass, it can settle, and after when it settles, it starts to build layers of protection.
So can you tell us a little bit about the difference between in vitro studies and in vivo studies? And then how often do we think the results of what we see in an in vitro study in the realm of Lyme, how often do we think those actually carry over into a more complex human organism?
Absolutely. And this is one of the things we try to move away from, because it's more and more evidence that the in vitro data cannot correlate well with what's happening in the body. So you heard, for that very reason, first we tried to do some mouse experiment, but it's very expensive. With small school, we couldn't afford it. So just very recently, we created a zebrafish model. And you're going to say, zebrafish, why zebrafish? First of all, it's very inexpensive. We can make as many embryos as we want. So we could have huge experiment. The recent data shows that if we want to eliminate Borrelia, we need multiple antibiotics, maybe different treatment. And it's only on one strain. How about the other strains? So we need to have something when we can test massive amount of different combinations, different novel antibiotics, maybe some alternative agents. So for that one, the mouse model is not efficient. So we are extremely excited. Just the last couple of weeks, finally put Borrelia into zebrafish, and looks like it didn't kill the fish, which is what you understand. The fish survived, and right now first question is any kind of, because it was embryo, so the first question was whether it's gonna become adult fish. If it becomes adult fish, where Borrelia will go. Do we gonna see any kind of morphological changes? What happens to the fish? So, but if we can establish it further, we're gonna have a model system when you can test all those antibiotics, all those combinations, maybe some crazy new ideas. But hopefully we have a system when we can afford to test multiple antibiotics in multiple species, and so on and so on.
Wow, that's exciting. So for those people listening, since these terms may come up again, so as I understand it, in vitro is what you refer to as the culture plate. So you have some Borrelia in a culture plate, you put some antimicrobial that you're interested in seeing how that reacts with Borrelia. And then in vivo is what's actually happening in the body, or the ability to do a study in an actual human. And
Correct.
So now you've moved from in vitro to being able to at least do these types of experiments in zebrafish, so that they're kind of in vivo, right? I guess would that be considered in vivo?
Oh, it's a vertebrate model organism, and,
cool.
and not just Borrelia. They tested other actually biofilm making pathogens in zebrafish very successfully. So we might have something which we really can use it for those in vivo studies. Now it's something between in vitro and in vivo. Actually, we also trying some small biopsies, skin biopsies. Right now, it's just from a mouse, and we try to see if we can inject into these biopsies some Borrelia, and we can study how Borrelia establishes itself in the skin. So that's the other thing we're trying right now. So as you see, we're moving away from the in vitro. I call the test tube studies. I think it's time to get something which is more resembles what happens in the human body.
Excellent. Yeah, this is really exciting. What role does Borrelia play, in your opinion, in neurological diseases such as Alzheimer's? And maybe you can talk a little bit about some of the collaboration that you've had in this realm with Dr. Alan McDonald.
Absolutely. I mean, he discovered back in 1990s or even earlier. He had this hippocampus cultures, and he found from Alzheimer patients, and he found Borrelia in those cultures. So since then, this is still very controversial. Yes, the whole infectious origin of neurodegenerative diseases. I think the strongest evidence we have right now, we presented in Finland, actually in one of the ILETS conference in a couple of years ago, when we had a patient who had Alzheimer and Lyme disease at the same time, and we found Lyme bacteria sitting in those Alzheimer plaques. Since then, I have another professor who is very interested here at University of New Haven, and we're collaborating, and I'm hoping that soon we can have more cases and can maybe provide more evidence for this theory.
And are you continuing to collaborate in any way with Dr. McDonald? Is he still researching at this point? Or?
I mean, right now he retired. He actively retired. The last I heard, he enjoying Florida and the retirement.
Well, he deserves it. He certainly played a big role in the history of Lyme. So you mentioned biofilms. Talk to us a little bit about biofilms in Lyme disease. How much of an issue do we think biofilms are in terms of persistence of Lyme disease? And then based on the research that you've done to date, are there any tools that you found that may be helpful in reducing biofilms in people with Lyme disease?
All right. So I don't know, that 2 years ago, we got a grant from Global Lyme Alliance, and it was a grant on a very famous case, the Ligner case. We were able to get autopsy tissues from this case, from different organs, including heart, brain, kidney, and liver. And we spent 2 years. We just sent the final report back to the agency, and we're working on a manuscript. We spent 2 years to find, can we find Borrelia in this patient? She died from Lyme disease after 16 years of treatment, multiple antibiotics, on and on. And the answer in brief, yes, we found in every single organ we studied, we found Borrelia, and actually mainly in biofilm form. What we also found is, because we were interested whether any kind of inflammation, any kind of tissue damage surrounding this biofilm in these tissues, and we do have evidence that is inflammation and tissue damage. So right now, and when I presented this in a couple of conferences, they said it's only one case. So right now we're collecting, hopefully the new biobank, we're gonna collect more cases, and we try to see if we can expand on this study.
And are there specific things that you think may be helpful in reducing biofilms in those with Lyme disease? Have you done any studies on agents that might help with reducing biofilms?
I mean, that we're looking for this now at least 10 years. So what we find is antibiotics has some effect on biofilms, but we didn't find it as effective than, like, for example, stevia, which is, I know everybody's why stevia. Something about stevia, when you have a biofilm, remember it is very, very protected with layers of different material. And the question is how the biofilm fits. Yes, if you cover yourself that much, it has to be somehow the nutrition has to come in. And we have some atomic force microscope images showing that there are channels there. So we were thinking that maybe this channel is opening up, when the bacteria sense that there's maybe some nutrition coming in, and let the nutrition coming in. Now, stevia being the sweet leaf, we're thinking that maybe tricks the biofilm, thinking that there's a nutrition coming in. In the meantime, stevia has a very strong antimicrobial effect. So that's our theory. Right now, everybody asking me what's the next step with stevia. I have a chemistry professor here who is isolating different fractions from the stevia, and we're gonna see if we find the active ingredient in stevia. We might not.
So I was going to ask about stevia in a little bit, but I'm going to go ahead and jump there. So one of the things I've seen kind of debated online about the research around stevia is the whole, test tube versus in vivo or in a person. And so my observation has been that when people take stevia, that they often do have a Herxheimer reaction. And in many of those people, it's very clear to them that it does seem to have an effect. I know many practitioners that are using it in their practice in obviously more complex protocols. But what are your thoughts at this point on the potential for stevia to work against Borrelia and Borrelia biofilms in the body? Do you feel pretty optimistic that it does actually have an effect in the body?
Of course, we need research data, but at the same time, I'm also getting emails from. I just got it just 2 days ago from this young person. She's still in high school, and she said nothing else helped her by stevia, saying that we still have to have some kind of clinical data or maybe the in vivo data from the zebrafish. I don't know. I can convince anybody to run a mouse studies on it. Right now, probably the easiest would be. If we can get some clinical data. Fortunately, stevia has a very safe profile. FDA cleared it. So as far as I know, Richard Horowitz is interested to running some trial. He has a small group of patients who is right now he's monitoring it with stevia.
And my understanding, if someone wants to incorporate stevia, obviously not suggesting they self-treat, but with the agreement of their doctor, my understanding is that it should be a whole leaf tincture, and we're not talking about the powder that you might get at the local grocery store to put in your coffee. Is that correct?
We tested whatever stevia we were able to get. You can now Truvia and everything what you can get from the grocery store. Tested something about the liquid form. A lot of companies now selling the liquid form. They work the best. Now, I saw actually one of the online comment that it may be the alcohol content of the stevia works, but we if you read the paper carefully, we tested the same amount of alcohol to be sure it's not just we seeing the alcohol effect on Borrelia. So that was controlled. That was a control study.
Plus, there's many other herbal products that people use that have alcohol in them as well. Absolutely. That don't have an effect against Borrelia. Absolutely. Or co-infections. Yes. So I want to come back to the persisters. So we talked a little bit about how persisters maybe are different from what we used to think of as the cyst form. What are some of the things at this point in the research that may be helpful in addressing the persister issue? So as practitioners continue to evolve their approaches, how does this whole persister concept come into creating a therapeutic protocol?
I mean, I think is what we found in this study with using the autopsy tissue for this Lyme patient. They are definitely biofilm forms in the tissues. So the question is, should we open those biofilm forms in a clinic and let everything out? I talked to a couple of clinicians and they said, don't open the Pandora box too fast.
I agree.
So I think that, and I saw those biofilm protocols on the internet, and I just want to warn anybody that just think about this. So you're opening a potential huge colony. Suddenly, all this spirochete gonna come out. Is it a good idea? So I think it has to be some very, the protocol has to be still established.
And kind of extending on that, so if we have these biofilms that are full of toxins and different microbes, and we start to release those, we then have the likelihood that the immune system will have some type of response that could lead to significant inflammation and significant Herxheimer reactions if it's not done properly, and if someone isn't also detoxifying and able to kind of get rid of those things in the body, right? So sounds like and I agree, it sounds like you're recommending caution. Biofilm treatment is not a do-it-yourself-at-home kind of topic.
Absolutely. And you're right. I mean, we still don't understand what kind of toxic material can come out from this biofilm. So we have to be extreme. This is don't kill the patient with some smart protocol.
Absolutely agree. So one of the first things that I remember reading from your work was the use of Cemento and Banderol, the two formulas from Dr. Cowden's work and Nutrimetics, and how those were helpful in dealing with Lyme and Borrelia biofilms. And I was surprised in preparing for this that was like 8 years ago already. But I'm wondering if you can tell us, what are your thoughts about your observations from that study in terms of using things like Cemento and Banderol in Lyme disease?
You know that in the last 8 years we tested other antimicrobial herbs. There are a lot of good herbs actually, which works against Borrelia and actually works better than the antibiotics. So, but again, I'm just gonna close this one. But again, that's very important that everything was test tube. So that's why we feel so strong about it. That's great that those herbs has effect on Borrelia, but exactly in a body, what's gonna happen is the herbs gonna reach to the site where this Borrelia exists. What happens in the surrounding tissue? So I think it's really have to move to the in vivo studies right now.
Okay. In your opinion, with all of the research you've done, in those people that are treated with antibiotic therapies and don't fully recover, is this an autoimmune condition or a post-Lyme syndrome, or is this a persistence of infection? This is just my opinion. I want to emphasize it. What I've seen in this autopsy tissue, I so strongly believe that Borrelia can survive, because what those layers of biofilms in the tissue, because we were sectioning the tissue and we saw that it's a very three-dimensional structure. To eliminate this, obviously, as we discussed, we need a very smart protocol, but I can see how Borrelia can survive it. Now, when I say survive, at the same time, obviously, Borrelia wants to spread. I mean, every bacteria wants to always grow and grow and grow. So I could see that some spirochetes coming out all the time from the spirochetes, which is practically stimulate the immune system. They try to go after the spirochete, and it creates a very, autoimmune kind of condition. So if you ask me, I believe that Borrelia is capable of surviving in our body and capable of stimulating the immune system for a long, long time. So it's actually a combination of both, but yes. The autoimmune condition, if it's present, is likely driven by a low-level persistence of some underlying microbial burden. Yes. And people ask all the time, can we cure Lyme disease? Can we eradicate every last Borrelia? My thought has been that we can't necessarily get rid of every Borrelia, but that lots of people have Borrelia and they don't have Lyme disease. So if you approach Lyme disease treatment with the idea that you have to kill every single Borrelia, to me, that's not very empowering. And I don't think that we really can do that. But I think lots of people do get well, do become asymptomatic, and they may still have some Borrelia in their system. So it's not necessarily the case that we have to get rid of every last bug in order to feel better and to have fewer symptoms or be completely asymptomatic.
I agree with that 100%.
Okay, perfect. You were one of the consultants on the Advanced Labs Borrelia culture, as I understand. And so can you talk to us a little bit about why is a culture a better test than an immune-based assay? And then for those people that suggest the culture may have false positive results, which I've heard, what are your thoughts on that? How is that even possible?
I mean, first of all, a culture is always a gold standard for any kind of microbiology testing. So, and not to have a culture for Borrelia, I think that's a crime. So we have to have a culture. Addressing the second one, that is a contamination. The lab right now has hundreds of negative controls, which all came back very strongly negative. So this is just a little. I understand what CDC tried to say. Unfortunately, we for some reasons we couldn't really send out the rebuttal, and I wrote the rebuttal. And in rebuttal, I mentioned that if you look at the data carefully, what they claimed is contamination was coming from two different sequencing data. And if you look at both sequencing data, you could tell it is not the laboratory strain, but some hybrid strain. So, by saying that, we need a culture test. So it's possible that it might need more work, but having just serological tests for Lyme disease is not enough, especially when we have a very low sensitivity test. So we need a culture test.
And so, as I understand, one of the challenges is in these immune-based assays, someone could have Borrelia in their system. It could be creating symptoms, but because it's so good at kind of hiding, changing form, it could just be the case that their immune system either isn't seeing it or is suppressed because of long-term infection with Lyme, versus something like the culture where we're not looking for the immune system's response. We're saying, is the invader here? And we're looking more directly. And so, I think a lot of times things like Western blots and others where people maybe have initially a false negative result, they start doing some treatment, they then have a more clear positive result. It seems to me that it's nice to have both an indirect or antibody-based test and a direct test, but not to rely solely on the immune-based assays.
Borrelia can do something strange to the immune system. You know about the whole seroconversion issue that people coming with IgM, which is always high, and no IgG, which is absolutely unheard from other infection. So Borrelia can do something strange to our immune system. This is one thing. Also, I just have a friend who just came down with IgM and very, very super high, and nobody believes that she has practically, there has to be some acute, and I keep saying that doesn't mean that just because it's just IgM, it doesn't mean it is acute condition. So we have to keep this in mind. Also, you heard about that some people has a very delayed reaction, even making the antibody. So this is a second layer. The other layers, those tests are based on practically one strain, a B31 strain. Only IGeneX has a test for 2 strains. I mean, how many more strains we probably have? 200 plus. And the Miyamoto not even testing with our surgical test. So there's so many layers of problem with that. So when we have a test like that, we definitely need some kind of confirmation. Now back to the culture test just for a second. The beauty of the culture test that we could see the spiroid even if it's not Borrelia or burgdorferi B31 strain or Borrelia Biofilm. So we can see other Borrelia strains, which I believe it is probably hundreds of strains.
Absolutely. So let's talk a little bit more about the IgM and IgG since you brought that up, because I think it's an important thing that people don't often understand in Borrelia. And correct me if my understanding is wrong, but in most conditions where there's a microbe that invades the body, IgM is, oh, you recently got this, it's an acute response. IgG is, oh, it's kind of a memory response. It's you had it in the past, maybe it's not a significant issue. And I even have heard people that treat Lyme suggest the same thing, that, oh, their Lyme was very new because they had IgM, or, oh, it was very old because they had IgG. My understanding is that because of the way Borrelia is so capable of hiding from the immune system, that you can actually, with the same exposure from potentially years ago, that you can cycle back and forth, IgM, IgG, IgM, IgG, that maybe the immune system stops kind of seeing it as an acute issue, so you become more IgG, but then at some point again, the immune system says, there's some Borrelia here, and it starts creating IgM again. And so to jump to the conclusion that IgM means it was recent, my understanding is that's not correct.
Yeah, if you go to any kind of ILATS or LDM meeting, always one question for sure from the audience about the IgM and IgG issue. So I have a theory for that. It's just a theory. I think that the fact that this IgM comes up even late in the disease, I think it's something to do with the biofilm. Because don't forget, so biofilm is covered with layer, but here and there sending out some little soldiers. I wonder these soldiers almost act like a new infection, the immune system thinking there's a new infection and try to create IgM again. It's just a theory, we have to test it.
Yeah, that makes a lot of sense. You worked with Dr. Ernie Murakami on the potential use of CBD and Lyme disease. And so what were your findings in terms of the potential benefit of CBD in those with Lyme disease? What did your research show?
I mean, he asked us to check it directly in a test tube. Obviously, CBD can have lots of different effects on the human body. So in test tube, we have seen some effect. The student who finished that study, unfortunately, graduated. And we want to confirm the data before publication. And for a year, I couldn't find another student. And just a couple of months ago, somebody came and wants to finish the study. So we're going to finish the study. The question is the CBD oil has any kind of direct effect killing Borrelia, or what you're seeing in a Lyme patient, because they do get benefit from it. Maybe it's helping the immune system. We don't know yet, so we're still working on it.
Okay, very good. And one of your more recent papers, which was fantastic, has been the use of bee venom therapy for Lyme disease. So how did you become drawn to explore bee venom in your research?
I mean, we tested over 100 different herbal stuff, antibiotics. So bee venom, we got a little bee venom, and of course it was a 1996 paper showing that bee venom, the melittin component, can have a direct effect on Borrelia. So I have lots of students who are very interested in this alternative antimicrobials, and one student really wanted to test it. So we revisited the paper. Fortunately we got good quality bee venom and good quality melittin, and from day one the result was very, very strong. So day one, the result was very, very strong, and actually she finished the study for a whole graduate thesis project. And I just only one comment on bee venom: it's a venom. So I know people are using it. I still want to be cautious about it because we still have to understand how to use bee venom safely. I mean, I don't know, that they use it for HIV therapy and cancer therapy, and they couple it to some nanoparticle to make it safer. Somehow it protects it from killing the red blood cells. Very interesting. So I think we still, if we want to explore this treatment, we need to understand how to use it.
So in the study, how did bee venom compare to the antibiotics that you also looked at in terms of its effectiveness against the various forms of Borrelia?
It was very, very effective. If you look at the graphs, practically the bee venom, we couldn't detect any live Borrelia, and the beauty was that even on a biofilm form, which is the most resistant form we found, was very, very effective.
And then how does the concentration of the melittin that was used in the study compare to what one might get if they were doing live bee venom therapy? Was it significantly more, or was it kind of in the range of what live bees would potentially offer?
Yeah, I actually asked a couple of patients, and I think we did some calculation, and it was in the range. So that's doable. Again, my comment is still just be cautious. It's a venom for a reason.
Yeah, absolutely. And I think, obviously, again, people should be working with their doctor. Nothing in Lyme is self-treating. I know I follow some of the bee venom groups as well, and they're always needing to have an EpiPen and do a testing. There's a whole host of things that have to be done. So please don't go running out and start stinging yourself with bees without being connected with someone who really knows
Absolutely.
what they're doing.
Absolutely.
What other substances has your lab evaluated that you feel hold some potential promise? So one of the listeners asked if you've done anything looking at the various silver products that are on the market, for example.
Actually, the silvers we tested very, very early, but that was one of the line, and I think they worked very, very well for the cyst form. But I remember at this point we didn't have a full line of testing for biofilm form, so I don't have the data yet, and we didn't revisit it yet. Yes, it has some effect. Again, I always try to say silver is a heavy metal, so you have to be careful, when you use it. But again, I heard that some people having very good results with it.
And are there any other substances that stand out in your mind, herbs or other things that you've done research on that are potentially of interest in people treating Borrelia?
I mean, we tested the whole line of the cardon herbs. We tested other herbs. But whatever people are using right now, we tested them. And I have to say that most of the herbs has good effect. So the oregano oil, probably heard, is just came out from the John Hopkins research. We also tested oregano oil, also find some good effect. So what we're doing right now, we actually retesting everything on biofilm, because after what we have seen in this autopsy tissue, we believe that's maybe the form we need to really address it. So we have some new techniques how to assess the effect of herbs or even antibiotics on biofilm. So actually we do a lot of retesting right now.
Yeah, and that's why I think it's interesting that you're starting to move into an organism like the zebrafish, because a lot of times with herbs, it's not so much that the antimicrobial properties alone are the way that it's working, but sometimes they're supporting the immune system, they're helping to support detoxification. There's so many other things that also have to be considered, as you well know. So I think it's really exciting that you're starting to do more work in that realm. Given all of the work that you've done in chronic Lyme disease, do you feel that antibiotics still have a major role, or do you feel that the herbs and natural substances seem to be a reasonable direction for people to consider?
I would do both. I find that some people really antibiotics works very well for them, and some people the herbs works very well for them. So I wouldn't just stretch the antibiotics. Yes, some people are combining antibiotics and herbal protocols, and with great success. Again, that's why we need a system, zebrafish, which can test all the combination, maybe combine the antibiotic. Now, doxycycline, for example, is a good example. The gold standard treatment for Lyme disease if you go to a standard doctor. Actually, on a spider heat, doxy has a very good effect. The problem it comes to the alternate form, doxy sort of failing. But if you combine, for example, we combine doxy with stevia with great success. So that's what I try to say is I'm not necessarily just saying just antibiotic or just herbal treatment. Whatever works. Whatever works.
Very good. Has your lab done any research in the realm of co-infections like Bartonella and Babesia? And if so, are there certain things that stand out as promising against those? My observation has been that Bartonella and Babesia oftentimes create as many or more symptoms for people than what Borrelia does. So what are your thoughts on co-infections?
We did a lot of testing in ticks. So I remember back in 2004, 2005, we went out, collected lots of this, over 5,000 ticks, and we looked at Borrelia, Babesia, name it, we looked at it. And I remember the reason we sort of stopped the study, because everything we looked, we found. So it's not surprising. This tick lives in dirt, so of course, it can be harbor all kind of pathogens. And now we have more and more evidence that with a tick bite you can get different co-infections. Now, our interest actually, we started to look at biofilm, vector biofilms, whether it is single species biofilm or multi-species biofilm. If you look at the oral biofilm research, it's always multi-species biofilm. So we went back to Borrelia biofilm and we started to see what else we can find there. So this is where our research is going, whether on what we're seeing under those layers is only Borrelia can be found, or maybe something else. I have a theory also for that. So when we looked at Borrelia biofilm, it's beautifully structured. It's like making the city with all the houses and all the structure, and after you can invite the people in. Yes.
You're probably the only one that would think of it as a beautiful structure.
I mean, if you look at those atomic force microscopy images in 2012 paper, it is gorgeous. Okay, saying that, we just about sending out a paper looking at again go under the structure, and we found Borrelia actually coexists with chlamydia. Now, not the chlamydia you think of, is probably a clinical species, but we find chlamydia in the Borrelia.
More like chlamydia pneumoniae versus
probably. We have some sequence data, and it doesn't look like pneumonia or Pseudomonas, but maybe some clinical species which can come from the ticks. And we were writing the manuscript, and just the last couple of months, more and more people finding chlamydia species in ticks. So it actually makes sense. And of course, you're going to say, why chlamydia? First of all, I remember I was in this European conference, and there was a lot of talk about chlamydia infection in Lyme disease patients. So that was one of the reasons we looked. And again, a lot of paper came out that in infected skin, when they find Borrelia, they find chlamydia. So that's one of the reasons we looked, we ordered some chlamydia-specific probes. And it definitely looks like chlamydia somehow coexist with Borrelia. Now, why do they coexist? When you look at coexistence of any kind of pathogen, they always do for some reason. Mm-hmm. One helping the other. So my hypothesis on that Borrelia having these beautiful structures, creating the structure part of the biofilm, and maybe, for example, chlamydia can provide, for example, iron. Borrelia cannot use iron, but chlamydia can use iron, and it was one of the papers showed that iron can help to make the structure even what we do, yes, structure even more solid. So that's where we're going right now.
So is your lab also doing testing for potential therapeutic interventions for things like Bartonella and Babesia, or is the focus on Borrelia at this point?
I mean, right now, focus on Borrelia biofilm, and focus on creating the novel model system, which I mentioned. So that's quite a bit of test right now.
Yeah, absolutely. I know you have been involved in research on Morgellons, and is there a connection between Lyme disease and Morgellons? And what are your thoughts on what's happening in those people that develop Morgellons? Do you think this is a Borrelia that's kind of the cause of Morgellons? Do you think it's potentially, I've read some people suggest Bartonella. What are your thoughts on what's happening in Morgellons?
Obviously, Morgellons, it is a very specific skin condition. So we have a couple ongoing studies right now. Obviously, we're working with Dr. Middlewin to find spirochetes, and we have a couple of papers with her. We found the spirochete in those tissues. However, you can always say, so is it a Lyme disease condition? If you see, not every Lyme patient has this condition, so it has to be something more specific. So we're looking for co-infections. So if we find something else than Borrelia creating these skin conditions. And I have a very interesting project. We had a chance to sequence 3 Morgellon patients completely, like we call the whole genome sequencing. So we have sequence data, and actually we compared their sequence data to some healthy controls. It was phase 1, and we tried to see if there's any kind of genetic variation in the genome which different from healthy controls. And actually we have like 9 very interesting variations. So right now is a study going to compare it to Lyme patients and healthy controls to explain why those people are having this very specific skin condition.
Very good. That's it's a condition that certainly people suffer tremendously from, and similar to how Lyme was a decade ago, most people don't think that the condition is real, and so I think that obviously will be proven that it is very real, and so I certainly appreciate people like you that are and Dr. Middlewin and others that are out doing research and studies in this area. How close do you think we are to cracking the code of Lyme disease? Do you feel optimistic at this point that we're finding better and better solutions for those people that are still suffering?
Very much, very much. Even 5 years ago, if you asked this question, I would probably say something. Yeah, we're working on it. No, I think we're getting close. I think that having John Hopkins, obviously, UC Davis, Harvard University, coming in and confirming all those research data we had it before, but now they're also saying it helped tremendously. The other is having those antibiotic studies, even in a test tube, showing it we cannot kill this bug, even when you be put right there in the liquid next to them. I think it was very eye-opening. So I think we at least we understand that we're dealing with a resistant pathogen. I think now the next step is just to find what exactly how can we address it.
So with everything that you know now, you had your Lyme disease experience years ago. What would you personally put in your top 3 or 5 list in terms of things that you might consider using to treat Lyme disease?
I mean, obviously we need some good antimicrobial. And some people like antibiotics and works for them. So I'm not going to say that only alternative works for them. So find the antimicrobial. I 100% agree with you. Borrelia makes some kind of toxic, some kind of neurotoxic. How do I know? I felt it. I've been there. I remember the feeling, how it feels. So some kind of detoxification protocol is a must. And I think the third one, I think, we are a spiritual human being. I think it's very important that you believe that you will recover. I think if you don't believe in it, not going to happen. So I think that number one, I would start that believe that you can recover, and we already have some tools right now. We don't need more research right now to get you better.
It's really interesting to me. I didn't know that was going to be your answer, but essentially you said the mental, emotional, believing you can get well, the detoxification piece, and then the good antimicrobials. And that's very consistent with what I started learning from Dr. Klinghart's work now, 12 years ago when I first started meeting him, and what he at the time called the Klinghart axiom, that we really need to focus on all three of those things simultaneously to make progress. And so, very interesting answer. So talk to us a little bit about how your lab is funded, and then if people are interested in supporting your work, how can they get involved? How can they donate potentially?
I have to acknowledge University of New Haven. Day one, they provided not too much money, but enough money that we were able to go to the woods, collecting some ticks, run some basic experiment, and start the research. And slowly, every year, obviously, that we start to have data. So I start to apply for first. Remember, Turn the Corner was one of the agencies supported. Lyme Disease Association supported us. There were small grants, but it was enough to get to the next phase. And of course, at one point we got the atomic force microscopy, which was great. LymeDisease.org helped us to get those Lyme. Global Lyme Alliance now from Turn the Corner also helped tremendously. It's amazing how sort of folded, through the years. Start with $2,000 budget per year, and now we don't have a huge budget still, but enough to get to the next level. University of New Haven also built a lab for us, so this is a full, beautiful lab on a campus for Lyme disease research, and they support the lab. And anybody wants to support us, is very simple. Actually, we go to the Facebook page. Every information is right there how to support us. And thank you in advance.
And the Facebook page is what?
Just go to Facebook and just put Lyme Disease University of New Haven, and it's going to show.
Okay, very good. And I'll find that and I'll put it in the show notes as well, so people can just click on it and find it also. Mm-hmm. So the question I always wrap up with is, what are some of the key things that you do yourself on a daily basis in support of your own health?
I take vitamins, not synthetic vitamins, but whole food vitamins. I remind myself back to the whole spiritual that, how blessed I am every day. It's extremely important. I do a lot of I should do more exercise. I believe that you have to move the body. And again, I'm very happy with my work. I feel every day I'm getting very, very beautiful emails every day, which warms my heart, how all research helped a little bit to those people to get better. So that's how I feel about the day.
I just want to thank you for taking time to be here today and share with us. But I want to thank you even more for living your purpose, living your passion, doing work that I can tell just from talking to you, it's so deeply meaningful in that your work is affecting lives of other people in a positive way. And I think that's something that, many people never get the opportunity to really experience their true purpose and passion. And I think you've done tremendous work. Like I said, I think I've known you for a decade at this point. And so you've definitely been one of those people in the realm of Lyme disease that has made a difference. It's nice to know people like you are out there continuing to research, continuing to try to find new solutions. And so I just want to honor you and thank you. It really does mean a lot to those of us who have had or are having a Lyme journey. So thank you.
Thank you, Scott. And I have to honor you because you also do tremendous amount of good for Lyme disease. Thank you.
Thank you so much, Dr. Sapi.
Thank you.
To learn more about today's guest, visit betterhealthguy.link/Eva Sapi, PhD. Eva Sapi, PhD is spelled EVA SAPI, PhD. That's betterhealthguy.link/Eva Sapi, PhD. You can also find her on Facebook at University of New Haven Lyme Disease Research Group. That's University of New Haven Lyme Disease Research Group. Thanks for your interest in today's show. If you'd like to follow me on Facebook or Twitter, you can find me there as Better Health Guy. To support the show, please visit betterhealthguy.com/donate. If you'd like to be added to my newsletter, visit betterhealthguy.com/newsletters. And this and other shows can be found on YouTube, iTunes, Google Play, Stitcher, and Spotify.
Thanks for listening to this Better Health Guy blogcast with Scott, your Better Health Guy. To check out additional shows and learn more about Scott's personal journey to better health, please visit betterhealthguy.com.